Testing. Oh, this is working. Okay. Making sure you're in the right room. This is application, health, disease. And if you're hoping for technology or with computational, you have to go to the sixth floor. But this is the cool session, so I hope you're here for that. Please come in front. This is meant to be a conversation. In the most ideal scenario, we wouldn't be using mics. So come closer. We showered. It's all good. Come closer. Okay. So I'm going to try to first set the stage a little bit about why we're having this conversation, right? We mentioned this morning we're still atlasing, we're still done with the versions, but we need to start planning what will be our next decade, right? And we want your input as we try shaping it because we're doing this together. And see, it feels very much unbound to think about spatial in the context of health and disease, and that's part of the challenge we have to think about today, right? And so what the exercise we're going to go through today is first, I'm going to try to break out the time. The first 25 minutes, really want to hear from everybody about what you think are the major, they go hand in hand, as Grace pointed out, and Kevin, challenges and opportunities that could really have an impact on our journey in terms of the spatial, whether it's for health and for disease, like big picture. And then moving from there, thinking about what are the immediate pains we could tackle together, what are the bottleneck? And then finish towards, from big picture to immediate pains, what could be minimal viable products we could work towards that could help move the needle. And why are we kind of taking that structure? It's because we're trying to fundraise. We're fundraising in a more centralized way to empower the whole community. And so we need to go from big picture to what would be the immediate, next step. And so we purposefully ask for Kevin to talk more about health and Grace about disease, because even though we've been doing a lot of healthy atlasing, obviously a lot of us are doing disease in parallel, and we should just embrace it because actually it helps us understand health when we also do disease. Okay, so with that, can you hear me? Huh? So with that, this is not an exhaustive list at all. This was just to get the conversation going in case this room would get quiet, but obviously none of you will be quiet. Can we see the slide? So, defining the scope, right? Let's start by defining the scope. Learning about the tissue structure, functional units, development dynamics in health, improving diagnostic prognosis, defining drug targets. There's so many things that can be done through spatial. So love to hear. We have some mics running around. I'm going to also run around. Big pictures. The challenge with disease is that it's unbounded. Like, when you think about a minimal viable product. So what are the major challenges in terms of spatial that you think HCA is best positioned into tackling? What should be our main focus?
I think disease really is an extremely large space, but there's one aspect which might be aligned with the overall philosophy of HCA to find shared physiology and pathophysiology. When you see how disease leads to organ dysfunction and loss of differentiated cell types, which are not disease specific, but are specific to the organ as it is challenged in its functionality. And that, I think, would align well with HCA's philosophy to float many boats.
So thinking about some shared functional units, and Matthias, I'm going to build on what you said, which is when we think about disease, a lot of diseases are systemic, and so as we're building our first version 1 atlas, we're going to be, bridging across all our organ system to have a global picture. So disease may help us think about more globally about this system, thinking about this shared structure, and I think it's a great idea. So Bruce, and I invite, of course, Kevin and Grace to jump in. Bruce. Is this on? Which color is your mic? Green mic, please.
Okay, there it's on. Unless you guys wanted to say something first before. I felt bad for Grace having to follow Kevin because that was so phenomenal, but then you were amazing too. And I think both your points were just on so many angles, so great. And let's say take one in particular, this mission to really have full connectivity between foundational image models with pathology, all the path reports, and then linking that up with multiomics profiling of spatial samples. So, I think it's totally feasible with disconnected immunopathology databases, but to get the best linkage, I think we have to make a much bigger investment in extremely high quality whole slide imaging that has all sorts of specialty stains in it, as well as goes all the way to the multiomics and 3D profiling. So with this, how are you thinking about that, Grace? I mean, you too, Kevin. But can we get HCA level investment in the, whole slide imaging, pathology, open up pathology departments?
Maybe,
slides.
Grace, before you jump in, I'll summarize, and I think it's a good point, which is when we think about spatial program, obviously we should be clear in that we're not sort of talking about spatial transcriptomics, we're talking about building an atlas in 3 dimension. And so this is about discussing what other type of modalities should we all be compiling, perhaps more systematically, especially since we're not exactly studying the same disease or the same organ, but should we also be thinking more in terms of measurements and the type of metadata that are tied to these measurements so that it would enable us to align. So for example, whole body imaging, H&E staining, and so forth. And I think this is an extremely important point that's being written, but any comments?
Yeah, I think this is really an exciting point. One of the things that we're noticing, so we have a large funded CZI project in which we're trying to link the cul-de-sac of the airway. So oral to nasal to trachea, tonsil, and even the distal tip of the lungs. And so one of the things that's been really challenging is each time we subdivided the lung, for example, into 6 proximal to distal units, there's heterogeneity that we're finding even based on anterior-posterior orientation. And so I think that one of the questions that I came into the HCA interested in from, that I give even to lectures to students when I do lecture, is like the 40 trillion cell argument, right? And then how many distinct cell types did we start with based on our kind of understanding historically? And then with molecular cues, how many do we think we are at now? Was it a couple hundred? Now it's a couple thousand, maybe it's 10,000, depending where you estimate. I think the concept of the functional units is something we need to start describing. How many functional units do we estimate in a healthy homeostatic adult there are? Because I'll tell you, when we start looking at disease, and we've looked at probably 7 or 8 diseases so far, the dramatic changes based on shared functional units is tremendous.
So Kevin, what do you mean by functional? I'm going to push on you. What do you mean by functional units? Because I think we're all like using this term and we all mean something slightly different.
Yeah, so for example, I think that, I saw Kirsten here, I think the functional unit at least of the glands, the periacinar immune niche that you described in the submucosal glands is a great example. There's immune cells unique there, different than the ducts. It's true we find, but slightly differently in the oral cavity. And so there's just ways in which I think that based on cells, molecules, structural ECM, all these things contribute to this kind of meta-level arrangement of what a functional unit is based on tissue niche and orientation.
So it's both a challenge and an opportunity. Where are you kind of hinting at every organ system, every bionetwork should start working in terms of defining their nomenclature for functional unit?
I think that would be incredible, yeah.
You didn't quite get to my point though of, I think that everybody that's doing spatial right now doesn't understand the value of getting an extremely high quality H&E sample that goes with every single spatial profiling. So we should have that as an SOP almost. And then, then the other opportunities, with biobanks and pathology departments, I think it's also something we should try to invest in.
I agree. I mean, I just want to add to this because I'm going to put my hat as a user for a minute and not as a moderator. I do think, I mean, it was very well exemplified through the lens of technologies that there's such a breadth out there. And if we have a challenge in aligning single cell suspension right now, there's been a whole different ball game with these technologies. So I completely agree in like a community agreeing on an easy, cost-effective measurement that all of us could do could already be a way of aligning, in addition to agreeing on a minimum set of experimental metadata schema that's well-defined and not too overwhelmed to accumulate so that, we could prevent having to work backward like we're currently doing. Completely agree, and that's something we should push for. There's a question here.
I was going to make a comment actually. We've got up on there kind of drug targets. I can maybe give some perspective from a drug discovery perspective of the type of data that we would like to see. I mean, obviously we've touched upon, and we've seen some great examples around really understanding mechanism of disease in the site of disease, cell-cell communication, etc. I think the other thing I think for us to consider is obviously a lot of diseases that we're interested in, perturbing with drugs or antibodies, are really quite heterogeneous, and so in that situation we really need to understand the different endophenotypes of disease. So you need large sampling, I think, across patient cohorts and really deep metadata that we've been talking about earlier on today, this morning, around those patients. So really understanding some of the clinical phenotypes, so we can really then start to map between what's happening at a molecular level to what we know from kind of clinical characteristics of the patients. One other thing, I guess, as well, as we start to have technologies where you can target your modalities more specifically to the different tissues. So, for example, oligotherapeutics, you can kind of get more specifically to different organs. I think important for that is understanding what are the cell type specific markers that we can go after to be able to target those therapeutics as well. And I can actually see a role for spatial technologies being able to, for us to be able to identify those types of receptors, et cetera, that we can then target therapeutics towards.
These are a really good point, and I want to clarify that HCA is not there to kind of help define all of the specific endotypes of disease. Obviously, we did talk this morning a little bit about HCA embracing disease more globally, and so we need to kind of figure out the right map because disease on its own, there's hundreds of thousands that they're unbounded to your point. But building a question, and maybe we get back to your point, is how do we empower this community to tackle any disease you like? Is there like core minimum sets of markers we can build upon the atlas we're building that one could leverage globally? And that's back to the point of like aligning data sets that you could take to figure out an atlas and then add your favorite markers for your disease, right? This is the type of thinking I think we need to think and align for. We can get back to this. I think you have the mic, and then, he's been waiting.
Thank you. The presentations were really nice and inspiring, and I'm just wondering if at the same time we're trying to focus too much on the taxonomy of the tissues. I think we're making the same, or we're following the same mental structure of going from classifying cells to classifying neighborhoods and trying to map them back to physiology, but the fact is that most of these diseases are systemic, right? And it's really unlikely that the localized neighborhood, I think the salivary glands, and there were great examples in which the structure and organization helped, but could we also try to think of other ways of using these single cell data, for example, coordination of all of these cell types, beyond cell-cell communication, like all of these cells work in community, right? Can we somehow link what we know from the past, like bulk transcriptomics? We can definitely generate all of these signatures, and they must point us towards more than a localized experiment. Like, making an analogy, if we are in this room, do we need the technology that measures every single molecule of air that is going on, or do we need to summarize it that we're having a discussion, we're having a conversation? Can we somehow also use the current atlases to try to learn these other levels of biology that maybe they are not highly detailed, but I would say also more practical to the clinic?
Can you give an example?
I would invite us to see the work that we're doing in the heart, but there, despite all of these clinical and technological differences, we do find a consensus multicellular response that in the clinic, when we present this to the clinic, they are really amazed because they expect that these diseases present in a very different way, but the fact is that they converge in an organization, right? And this is something that then we can trace back to bulk. So now we have evidences of large cohorts that have this coordination, right? And I think with Jose also, we've been discussing a lot the differences between compositions and organizations, right? Like, do we need to measure how many cell types of a kind you have in a tissue or how they talk, right? What we see is that different compositions lead to the same disease and the other way around. So I think it would be interesting also to put, whenever we start discussing disease, that it's not only the compositions. I disagree with Aviv that it's just a matter of compositions and finding the disease cell, right? Like, we do find them in, like going to bacteria, like all of these things of persister cells in cancer. There are many examples in which we can think of higher levels of biology rather than just classifying what we see in tissues, right?
So what would be your main recommendation here?
I would say to try to measure other, like, coordination, right? Like, I think there are many examples also coming from them to try to make these convergence. Like, convergence is not a bad idea, right? Diversity is cool, but all of these cells are working together and do the same thing, right? We have conserved developmental, like, programs.
So they're finding convergence, some of their redundancy in their processes. And you made a comment about systemic disease. I mean, without simplifying the issue, there's obviously some that are very more organ specific and some that are very much systemic. And so kind of there might be some different framework that needs to be tackled in these two contexts. And yeah, I think it's an extremely good point.
I just wanted to reflect on some of the points that have been raised with some perspectives that I've had. The first thing is, when do you bring disease? And you have to ask the question, are you bringing disease to solve the disease? Or are you bringing disease partly to actually contextualize reference? Because the approach to do that would be very, very different. On the one hand, you just kind of like need multiple states of disease to sort of like put into context what you find. But in the other, you'll have to think about carefully curated patient cohorts. And it doesn't just mean numbers, because that's just like a sledgehammer approach, but looking at, you know, putting patients with specific characteristics so that you have homogeneous populations that you can compare across, rather than having a range of somebody with disease X from mild to severe. And of course, you're going to need thousands if you're going to get rid of any noise. And then there's just so much noise that it's more complicated. So what exactly do you want the disease for is number one. The second question is, if there is any effort to look at disease, then I wanted to bring back the point that Matthias Kretzler made in terms of having public engagement and patient involvement in terms of what is actually important for people. And if HCA is going to go down that route, we can't just do that by deciding what disease we kind of want to solve or help solve or whatever. So that's the second point. And then the third point is, what can you actually, so 3 points, and I'll stop after that. The third point is with regards to the functional properties. I think, Kevin, you mentioned functional units. And I think we're at that stage where it's a good aspiration to have, but I am also kind of slightly skeptical how this will be interpreted and how this would, how it can be solved for the different, you know, tissues, etc. is useful, because all of these kind of how the cells come together to form a tissue and how they assemble together to form an organ are all really important questions. But what exactly is a functional unit? And that, you know, is difficult. You know, if you build on what HCA did really, really well, which was kind of like just help everyone use, you know, enable them with particular sort of like technologies, generating high quality data, which then made SES and assembling it into Integration Atlas. Where do we go next in a step-by-step manner?
So I think it was really helpful, like, to put these comments to context. Was very much through the challenge of mentioned in number 2. So that would be, how do we define an MVP for minimal viable products across organ system in the context of health and disease? Should we focus on any specific organ or bionetwork, specific disease? And how do we coordinate with, you know, under consortiums? Because there's obviously ongoing work where across consortiums that are disease specific, that really think about these endotypes, that really think about these in-depth characterization. So how do we align? Is it in terms of like the standards and the measurements so that it can eventually kind of connect? Thoughts as we circulate the mic?
You know, I think, you know, to people's comments about this, about, you know, also about the functional units. How do we define a functional unit? At what spatial length scale is it meaningful to define a functional unit? Is it literally a tiny niche within a tissue, or like, you know, interpatient or across organs? I mean, to a certain extent, and you know, this is my naive take, because you know, I'm actually a colorectal cancer researcher masquerading over here as a single cell person. That, you know, I think for something like CRC, which, you know, mostly concerns very specifically the colon, and it makes sense that that the thing that I care most about is the colon. Obviously, there's like some knock-on effects on other organs and stuff, but that's the main organ that I study. And maybe, you know, I don't know about the scope of the HCA disease program, but there's some work that needs to be done about classifying diseases based on, like you said, like which organs need to be looked at, and then what kind of like infrastructure needs to be built for those different things before like a decision needs to be made. Because there's some world where cancer researchers just want to maybe use some reference spatial atlas to make some queries about how some spatial structure that's quite local becomes disrupted in tumor. But then to other diseases, maybe it's more meaningful to be able to learn about the coordination between tissues.
Quickly want to address scope of disease, because it's the first time we start discussing disease in the context of HCA, and this is part of what Muz was mentioning, is that are we using disease to understand the context and the boundaries of health, and from which angle are we looking at? And you're going to hear more about this tomorrow's morning session when we talk about diversity and equity, and it came out loud and clear from that community that if you really want to achieve equity across different regions of the world, we need to consider disease, because in many parts of the world, they get mostly funding through the lens of studying disease. And if you want to show impact, we need to start considering this through different lenses. Of course, we haven't fully defined the scope, and this is partially what we're discussing today. What is in scope, what is out of scope? I want to hear your thoughts, right? So this is why it's not so perfectly bounded. So we have a bit of a whiteboard. It's like a white. And so there was a comment back then.
Chloe,
and
just
yes,
before,
yes,
I think
please.
there were some great points raised by Muz. I just want to, open up the discussion that I think she kind of got to. You described some of the excellent origins of the HCA. And one thing that we're noticing that has kind of been a philosophical question that we haven't answered yet is that when we use same slide multiomics, going to define cell type and weighting protein versus RNA to give cell type identification can be quite challenging. In fact, we see some cell types that we would call T cells based on one modality and not versus another modality. And this is across a lot of our cell identifications that we thought were pretty hardwired and pretty easy. And so I think HCA did a fantastic job of kind of originating for us, at least in our group, how it is that we can methodically proceed forward with community annotation on what a cell type is. And I think that the spatial technologies open us up as a group to start to ask that, both in a convergent context, fibroblasts across the body, but also in a multimodal fashion, that I think needs to probably be a discussion point in the very early status before we start thinking about functional units or disease, because it's really an original question that spatial opens up.
Yes.
One of the questions I have is how are we going to harmonize language between what we do as single cell biologists and what pathology is currently doing? And I come from a cancer background where it took us a solid 10 or 15 years to go from this is what we look at on a slide, it's DLBCL, I don't care anything else, to here is a molecular diagnosis of disease, and it took a long time to get that language shared. What's the plan?
So, what are your recommendations? We're discussing here what could be. Obviously, we're not going to solve it all within the next 30 minutes, but this is what I want to hear from this. Like we are asking about the big picture questions and big challenges which you're mentioning.
Yeah. Are we engaging with pathologists? Are we engaging with medical groups as a part of this meeting, or are we all single cell biologists who are accidentally bubbling ourselves? Yeah, how do you link them? Yeah.
I think I think these are important questions, and you're saying you should think more broadly about the expertise we're bringing in, and I think some of us definitely are working in pathologists, but we should do this more systematically. Actually, more importantly, we should make sure we engage with if we're going to go to, of course, indefinite disease, and we pick specific disease, having the right clinical expertise. And I know there's actually a few physicians in the room that actually are also part of the journey, and some pathologists as well. But yes, you're right. Yeah, there's a few, and then I'll get to you. I think there was like here. And then
Sorry, I think continuing on the same theme, in the digital pathology world, the collagen fiber orientation is becoming a common thing now for the disease prognosis and disease outcome. So that is something which can be easily incorporated along with the H&E to make it more equitable, because those are simple things to do. Any country can do it. If we can link your spatial data back to the simplest thing anybody can do anywhere in the world. That's one point I want to raise. The second point is the most of the cell-cell interaction now seems to be focused on 2 cell types. I work on a unique disease called inflammatory breast cancer, where based on this single cell data, it looks like more like a triple interaction, epithelial cells, endothelial cells, and fibroblasts. Probably we should be thinking about expanding the technology to see whether we can capture those complex interactions.
I just wanted to make 2 points. The first was with regards to disease. I'm a clinician, so I'm totally into doing disease, but I just want to kind of make people think and consider if it is disease, then it has to be done, carefully and engaging the right communities. And we talked about pathologists, etc. One of the things I noticed from the first time the HCA inaugural meeting was there were just so many people with different expertise, and including clinicians. And I remember raising that we need to bring pathologists and make sure we've got pathologists embedded in there. And aligning the histopathology is really important, this kind of like orientation, etc. Because one of the biggest criticisms, and coming from a clinical background, is sometimes we are using fancy technology to show something that, pathologists have known for centuries. Yeah. And it really does put them off, which I totally understand. And I think, there's no need to kind of have a complex algorithm that tells you what they already know, but actually leveraging on that, insight and knowledge over the centuries to kind of like make the actual progress. Those were my 2 comments.
I guess to just add, in terms of the disease, why we also mentioned disease beyond equity is that we realize a lot of groups around the world are doing both health and disease, and they're just saying, okay, well, I guess I'm contributing health and disease is going elsewhere. So should we just kind of embrace it more systematically? And so some of it is not so much starting new disease endeavor, but more embracing what's already being generated. Before we go to the, Kirsten has been waiting, and then we'll go to you.
Great, thank you, Chloe. So I've got 2 points. First of all, towards the pathology, I happened to go to the European histopathologist meeting last week, and actually I would say a good third of the talks were about single cell, and actually there's also maybe something this community can learn from them, because I think they've been in a way more advanced in using image AI analysis in their cell type annotation, and there were great examples how this sort of pathology AI can inform cell type annotation that then improves the single cell annotation. So it's a, it goes backwards and forwards, but I think those interactions are beginning to happen. So, but I completely agree that it's really important. And then the second point I was just going to make is around what can we really do as the human cell atlas. And I think, there are so many diseases out there, maybe, we have to have some sort of focus, and that still, I feel, has to be in the space of health, but in the context of disease. And personally, I think one of the areas that is very exciting is what Grace talked about, and also where the Teichmann has looked at gradients in tissue, so that we really understand the changes across a tissue and then maybe map disease at one or the other end of that spectrum. And again, that's where the spatial, I think, can really help.
So I just wanted to make a comment to what Maz has said, but really Kirsten addressed that, but I'll reinforce it. I think if we discover something that the pathologist already knew, it's already a good step, because it means that the technology is not finding something wrong. But we know that we can add on much more information on that. We can say what the cell-cell interactions are. We can start to predict drug targets. And what we can do is, like, if we think about drug targets, we have maybe made the mistake, or maybe not, but of thinking drug targets as very often as organs as a whole thing. But if we look at the heart, for example, and you look at some areas of fibrosis might differ even within the heart, what is the fibrosis that you really want to reduce? And there might be some areas of fibrosis that you need to keep, otherwise the heart will break. So understanding those, we can call them functional unit, or we can call them domains. It's very, very informative for the ultimate goal, which is the one of understanding drug targets. And I think that yes, in the end, we want the whole organ to work better, but in some diseases, you need to look at where is the seed of that disease. And if you think to cancer, the initial cancer is probably one cell. So the resolution of the disease varies according to the pathology and the history of that disease. So very, very hard to generalize here, I think.
Do you think there's some context in which, so I think you're making a very important point, and so to take a few steps back, we should definitely think about concepts that are more generalizable. Every bio-network can go and do their own deep effort, but as we try to build a special, I'll get back to Matthias, as we try to build a special program, maybe perhaps focusing on these kind of core shared principles is perhaps what you're hinting at. I don't want to put words in your mouth, but yeah.
Really following up on that, really, what I often refer to is the tragedy of the commons, meaning HCA is very good in finding what is shared. This is what we have started out with. And these usually are the things investigators, like we do in some of our rare disease networks, don't see. But those are the solutions the globe needs, particularly in low and middle income countries, that we can find solutions for problems who are shared and can address many diseases in common. That's the abstract common. The specific one is, yes, exactly. If you have a state where specific fibroblast activation is associated with rapid loss of kidney function in viral myocarditis, then that might give you exactly information which you can carry across organ function effectively. If, and that's a big if and the need for HCA, if we have set up our data capture instrument that they allow that crosstalk. If we set up our spatial platforms that they do have the same marker enabled in each of the different elements, if we have set up data sharing instruments, if we have set up clinical phenotypes who are capturing rates of progression in a meaningful way for that specific organ, but are also translatable.
So you just went through a slew of very important recommendations very quickly. So I hope I hope they captured them all. But you talked both in terms of these key technical metadata we need to collect. You talked about patient characteristics we should think about defining that go across disease potentially, and some of these technological approach. I mean, I think as we think about disease and things that are generalizable, I mean, taking the fibroblast, for example, definitely see fibroblast in many different contexts. So there could be also an angle, just putting it out there, common disease mechanisms. Definitely in the world that I live in, which autoimmune disease and immune-related disease are definitely common mechanisms out there. So I think I'm encouraging everybody to think more in terms of commonalities and not so much in terms of like very specific disease processes, which we're definitely not set up necessarily to do in this context. Any other, and I saw a hand, but thoughts here while?
I'm thinking a little bit about molecular and signaling gradients to some of the points that were made. Because you talked about drug targets, right? And this is of course something that comes out all the time. You have things like MET-PK inhibitors or like different kinds of drugs that target very specific signaling pathways. I'm only familiar with CRC, so I can only comment on that. But we do see that certain signaling pathways do have specific spatial localizations. I don't know how translatable this is across diseases, but I feel like it must have important implications for treatment. Even though I cannot say more. I don't know more, yeah.
I'll give an example just in the context of the shared diseases, but also some of the challenges with that. So because our bio-network had the opportunity, but also the challenges, just kind of in the theme of today, of incorporating disease as part of our atlas, when we worked with the group to establish both Tier 1 and then subsequently Tier 2 metadata, not based on the technical data, but on the clinical data, this threw a huge wrench. In fact, we actually had to engage a number of clinicians and pathologists to see exactly what metadata that they wanted as part of every standard input, both spatially and single cell, to ensure that those analyses, which are required at scale to make any real inferences, were all collected. And of course, because the oral cavity has these disparate functional areas, teeth very different than tongue, I had to engage clinicians of every type. And so this took a long time. And so one of the things that the HCA can start thinking about now is if we're going to bring on clinical or pathological partners that have clinical associated tasks as part of their workflow, we need to start engaging them now without doing it after the fact, because that post hoc analysis took something that we thought might be a couple-week task and pushed it to be a 9-month task as we start to engage not just our basic science, but our clinical colleagues.
I think also like mining existing FFPE samples, which is what everyone's excited about now that we have access to FFPE technologies, face the same problem, except that metadata is like buried in like years old written text form, which is like so difficult to work with.
Yeah. One thing I'll also add is that once we got this established, is that we know that across, for example, the airway is an example for us, that sarcoidosis, which is one type of disease, can show up in the lung. It also shows in the oral cavity. So in the context of Muz's question, we were going to use that as a way to further not establish solving the disease, but really kind of leverage the idea of what HCA does very well, which is as we start to bring together this integrated object, both single cell but also spatially, how can we ask questions about the manifestations of sarcoidosis in different tissue niches to understand how that biology is both shared and unique?
So we'll go here, but before we go, just remind the scope of the conversation, right? Obviously, every bionetwork should think about their roadmap and what they want to focus on. And here we're talking about where globally HCA should spend time and energy, fundraise to kind of empower all of you to do this at scale. And we'll start thinking about some MVP in about 10 minutes, but just to kind of help frame the conversation. So there was one in there, then it's you, then it's you, then it's you. So it's your number 2.
So we keep talking about a lot of different diseases, but we're getting away from the diversity aspect. And I think one of the things that we should think about in this context, and as you were saying earlier, ma'am, in the white shirt, maybe selecting a disease or two and then finding the commonalities between different diseases within that. Does that make sense?
Yeah, so there's definitely work being done, and you're going to hear more about this tomorrow, where we're thinking more about global disease burden so we can move the needle in terms of equity. But we don't want to say, we only want these specific diseases. Every disease are welcome, and of course, the community is already studying these diseases, and so we should fold in these data sets as it can still help us understand health, right? So it's not like we would necessarily say we're just going after 2 or 3 diseases in death. We're saying how do we balance, what type of information do we need to start folding in additional data sets that are being generated outside the scope of HCA, right? Yeah.
I kind of just wanted to make a suggestion of one possible option going forward that may be achievable and also targeted. And it incorporates several of the things that we've discussed in terms of making sure that we've got clinical expertise, bringing in pathology. And there are lots of tissue biobanks, pathology biobanks for a range of diseases. And I think, Grace, you brought up the point as well. There are a lot of technologies now that are very amenable to analyze FFPE, cells from FFPE, as well as tissue sections and so on and so forth. So this is quite a good kind of baseline, perhaps, to think about. And you can also achieve the diversity if you kind of make sure that the biobank, these tissue biobanks are, representative around the world, without actually talking about disease specifics, just the fact that, how about approaching pathology biobanks around the world and bringing those expertise and now deploying single cells so we confirm what they found and they know and we add much, much more.
Isn't it interesting that everybody's jumping at disease? No, nobody wants to do healthy. We have here, and then somebody else.
Yeah, I like the discussion on disease and health. Is something we do all the time in our case. But I think what we call health is also contextual. So for sure, one of the biggest issues right now for HCA is diversity. Yeah, and diversity comes in many different flavors: genetics, exposure, the environment. I'm an immunologist, when I look at a health control in Africa, for example, I'm based in Malawi, what I see in health control is not the same what I see in health control in the US. Yeah, the person is not diseased, they are healthy. So I think one of the things we should be adding to what everybody else's discussion is really putting an effort on going to places where there's different exposure from Europe and the Americas, yeah? And one way of doing that, I think Mars mentioned something, which is, it's very difficult to me to just go and say, okay, I want healthy individuals, and I put a classification of what I want, and we collect samples, and off we go, we create an atlas. Later on in life, it will help you. It's difficult to justify that, because there are diseases people face on a daily basis. So you have to do a balanced approach, where you study some disease elements, but also learn the health individuals in that population to develop the health atlases. So then you're building equity and diversity at the same time. Thank you.
Thank you. That really helped frame the conversation. Please.
So I am currently working on an analysis of fibrotic diseases across organs, and there are a couple of things that I'm missing in many data sets that are published currently. So one of the things I'm missing is a clear definition of disease. So oftentimes when people upload their data, they just upload the data with a certain diagnosis, but it's very unclear kind of how that diagnosis came about. And I can imagine it's very similar to the things we struggle with, for example, with cell typing. Just because one study calls a cell a myofibroblast and another study does too, doesn't mean they're talking about the same cell. And now we're coming up with definitions of cell type markers and so on. And I think this is also very, very important in disease. So we need a kind of standardized description of disease and phenotype. And then the second thing I'm really missing right now is the description of the severity of the disease. So again, just a diagnosis isn't enough. Some people have very advanced disease progression and some don't, and grouping every patient together doesn't always make sense. So it's really important to standardize how we describe the disease in terms of progression. And then thirdly, one other point that I think is often neglected is to basically standardize a way of how we describe where exactly the sample came from that we took. And what I mean by that is some diseases are very localized, and just because a patient was diagnosed, but then the sample may have been taken 5 centimeters away from the exact disease location, and that obviously makes a huge difference. So this is super important, in my opinion, to really annotate these samples very, very carefully in terms of these 3 points.
Thank you. So what I'm hearing, to summarize, is that the same way for the healthy reference, we're spending a fair amount of time thinking about these biological metadata. It gets even more complex in disease. And so both in terms of where the sample came from, which we try to do in terms of atlas assembly, but also in terms of disease. And so we should reinforce some standards if we go, I'm just summarizing, we should reinforce some standards if we are to move more systematically to disease so that we can contextualize these results.
Can I add to that? I think between those two comments.
Oh, you're behind me.
So as part of our pediatric efforts, one of the things that we did was, it was all supposed to be health, but we actually had a histology team come in and grade each individual sample across the entire FFPE section. And then there was an ordinal scale, so it went from truly, non-inflamed to actually almost looking like disease. It was so inflamed. And they all got a grade. And then we didn't know what to do with that, because these were healthy samples that were input, but regionally, 6 regions taken from each individual lobe of the lung. So again, 5 lobes, so there were 30 samples per patient, and there was a diversity across each lobe that was taken. And so the input on health was very unique for us to consider, firstly. And I think that the same thing applies if we start to add disease, is that I think there's an opportunity for HCA, no matter what the organ system, whatever the original, we probably need to come up with some SOP for how it is that we input samples as part of the healthy reference, because we'll always have the opportunity to take that first FFP, have it graded before it goes on to the technology that we want to profile with it. That kind of, I think, strictness will probably improve the quality of some of the other things we want to do downstream. But it really kind of adds this interesting flavor, which is, well, what do you do with the stuff that's in between that's not disease, but it's not healthy based on the original approach?
Yeah, we have been talking here, Maria and I, for the whole time, like discussing everything. So I'm just gonna mention some things, right? And one of the things about inclusion and diversity, I think we work in, I come from Latin America, and we work like everybody else. We do not send out a project about healthy tissue. We get grants for disease, and together with that, we get a healthy tissue. So what I believe that you said, like many people are generating data for diseases, and they're just not feeding HCA with that. So maybe a platform that is a little bit more fluid, that you wouldn't have to decide about a disease or another one, because even, well, in Latin America, we also have all the same kinds of cancer, you know, and they could be different. So it's not like you're not gonna be targeting middle income or low income countries by just choosing dengue or something that is more specific, right? So maybe not choosing a disease would be a way of like integrating all these data sets, and then maybe focusing on something after you get enough information, or maybe specific grants, or that you could say, okay, now we have a grant for a specific disease that it's like locally interesting, and then that would like make people like maybe feed into that more. But choosing and picking a disease, I think it's very, very complicated. Also, something that everybody's discussing here, what kind of metadata? That's an infinite discussion, and we have that kind of problem with all cohorts, like for everything, and a lot of effort. So I think it's very complicated to make this decision of picking a disease. And for inclusion, I think we would include every country. It doesn't matter. Like if it's fluid, they would be included anyway. So I don't know. We've been discussing a lot of that.
Thank you for coming. So to contextualize, right? And it's something that Ms. said. We integrate all disease that are already happening without us fundraising specifically for it, but it helps also contextualize human health and gives, you know, context through different lenses. And so becoming a little bit more flexible, yet defining some core sets of metadata so that we can figure out how to align it. So it's still embracing some of the technical metadata, which we haven't covered, but I think track 2 is also thinking about some of these metadata that are actually needed for integration, is your comment, as opposed to kind of picking one or two diseases. And I think these are important comments. We're not necessarily lobbying for 1 or 2 diseases. I think there's different tracks, you know. There's definitely like just not ignoring what's already being generated, perhaps embracing this as an opportunity, and then thinking about what's being neglected, perhaps with time, that we can empower. Yes, sorry, yes, go ahead.
No worries. Hi, I'm Tala from New York University, Abu Dhabi. So I wanted to touch mainly on the diversity aspect, and someone was previously mentioning that essentially it's our genome and it is the environment and it's this entire context in which where shapes kind of where disease is going and the susceptibilities and the severity. So I was just wondering, I'm new to HCA, like how much of whole genome sequencing or genotyping are we taking into account into shaping what is metadata to kind of put more into context the origin of the disease and molecular pathogenesis of disease and what makes us different across the world, essentially.
I'm looking for a representative of GDN. We have a whole genetic diversity bionetwork. Going back to Patricia, do you want to comment? I mean, I can certainly comment that there's actually a whole bionetwork that is very much focused on that question, the genetic diversity bionetwork. They have a whole session tomorrow morning. It's not every bionetwork that is mandated to genotype or sequence their patient, but there's actually a whole bionetwork that is mandated to do so, and it is through the lens of diversity. Of course, if we tackle disease, that could certainly be the same way we talk about H&E, which is a bit more cost-effective than perhaps sequencing, is something that could be recommended to try to help align. Yes, for disease, like thinking about the genetic underpinning, where specific disease definitely have genetic underpinning. So you're making a recommendation in terms of this, you know, a different type of additional data that could help contextualize. I think that's a very good recommendation. Yeah. Yeah, of course. Yeah. Thank you, Kristen.
Maybe just a quick comment on the previous person sitting over there. I agree that we shouldn't, as HCA, shouldn't at all tell others what diseases to look at, and really that's going to be a bottom-up thing, that people will be interested in their own diseases, and then it'll evolve. A lot of HCA has been very bottom-up, and I think that's worked very well. But I think what that comes back to is that what we talked about this morning is the infrastructure for putting data somewhere, having it in a format that it can be reused and put sort of back together in different ways is absolutely critical. And it's not always the most exciting from a biological point of view, but you know, a lot of the data storage and how to facilitate that and access is going to be really important to allow disease to be integrated.
To your question about sorry.
No, go ahead. Okay. Sorry. So just bring together two things. We were talking about ground truth to be learned from pathologists, and also about metadata about region side and about the severity of disease. And I think that's two things that are exactly what a pathologist does and has been established. So if we're looking for SOPs, they have been written, they're probably out there. So we just need to apply them. And secondly, I think one difficulty with disease, like when I would normally look at disease, it's often a systemic thing, especially if immune cells are involved. And then one problem we might face in some of our single cell efforts, especially if we're so organ focused, I mean, for us, for example, in the heart, that you could kind of lose track of the systemic production of some kind of cytokines, hormones, anything that reaches the heart. And you're looking specifically at all the expression you find in one organ, but then you're kind of missing the big picture of anything that is produced somewhere else, or cells that are originating, for example, in the bone marrow are activated in some kind of lymph node. And if you don't have matched samples that bring those things together, then you're basically missing the systemic approach.
Yeah, the question about the diversity, the single cell atlas that we recently published, we made sure that all the samples are genetic ancestry mapped. All the data are classified based on the genetic ancestry. And this genetic ancestry information has led to a clinical trial based on the genetic ancestry dependent differences. There are quite a few clinical trials in our place, which is based on the genetic ancestry. We're looking into the neuropathy associated with the genetic ancestry. So there are a lot of focus going on taking genetic ancestry into consideration.
I wanted to ask a question to the group in terms of, as the HCA is moving to integrating more disease samples, and the fact that there is this challenge that patients expressing that disease, there might be a lot of heterogeneity in their disease and how advanced it is. And so is there an opportunity to, with the HCA, as you're uploading this data, to also upload other medical records to kind of integrate more that type of data and generate more insights?
So I guess I'll quickly comment on that. We, of course, commit to open data access, but we have been developing managed access system over the last 7 years, which we discussed this morning. I don't know, we could release all medical records. There's obviously limitations that are institution specific, but the groundwork has been done so that such managed access information can be done, and then it becomes very cohort specific.
We'd probably need a platform that has a specific compliance and privacy.
So that exists. That's what we've been working on for 7 years. I mean, sharing medical record is it's a different type of challenge that's going to be very institution specific, and so it's a bit of a case by case. Bruce, and then I want to get us to the last 20 minutes to get a bit practical and pragmatic in terms of how we can move the needle. Like, go ahead.
I guess I just want to point out that when a pathologist when a decision is made to take a sample to send to a pathologist for them to interpret it, they're not just looking at the sample in isolation. They're looking at the entire clinical history, state of like why was the sample being obtained? What's the question to them that they're supposed to answer? So they're putting together a unified view of the clinical state, where and trajectory and everything is sort of included in thinking. They could be wrong and they could miss things, and and still, Kevin, even with this like nil to significant inflammation or fibrosis being present in the sample, it's still classifiable as a normal. So when you put that, maybe, at least at the phenotypic level, maybe. But it's all part that, sort of builds up this model. And I think what, Grace, you were saying about the power of these new pathology foundational models and Breakout and the others is it's extraordinary, and it's not like we need to do a deeply controlled study. The pathologist can make the call with an N of 1 situation, and we can teach AI. AI is able to do that now. So we need to be able to develop a power platform that stacks together all these data modalities such that you can enter with an N of 1 and get a meaningful interpretation of it if you supply, contextual details about like what's the magnitude of the, so let's say it's cardiac fibrosis or pulmonary fibrosis. What's the actual clinical phenotype? What are the PFTs? What's the ejection fraction? So it's an in-context set of metadata that has to be in there. I think HCA supplying infrastructure that could collect in-context metadata. We can learn what from a lot of AI approaches, from a lot of pathology reports. What's the metadata that's in context that's likely to be relevant without going through 9 months of interviews with docs? We can at least front load some of that data infrastructure and now put it together, give somebody something in sort of an organized package per domain to then call down from and agree to this as a minimal viable metadata model.
So to kind of summarize it, essentially we've heard a lot about metadata and context is really key, and think about that infrastructure. We only have, this says 18 minutes, but I actually know we have less than that because the keynote speaker is starting 15 minutes. So there's another hand. Is that a shortcut? Because I want to get us to something very pragmatic.
It's a MVP question. Is the fact that we have the schema and the ability to upload the data, does that represent a minimum viable product?
Yes. So thank you. Yes, but it's not trivial, of course, because every disease, every tissue is a little different. So I think also it's been interesting for me to. So I'm part of this task force, starting to think about this, and we'll try to summarize some of your thoughts, and there'll be more opportunity to connect and give more feedback about this as we try to think about the big pictures. We talk a lot about the big picture, but also the immediate next steps. And it's been interesting for me to listen to the conversation evolve, and almost all of you jump to disease right away. And I'm like, hey, of course, and we know normal tissue is being done with disease. And this was mentioned by Patricia and others, but it's very interesting that you just all focus on disease, which is hard to develop metadata schema for the reason we mentioned for every specific disease. But I think thinking about this metadata, about how the samples is collected, where, how, in which context, definitely a minimal viable product. Something else that actually I put up there that we've heard through some early bionetwork coordinators discussion is, this morning you heard about version 1 Atlas, right? Over the next 2 years, we're trying to kind of finish it, single cell suspension. And there's an opportunity there, we also heard, to frame the conversation, that there's like a dozen, if not 20, if not more, spatial technologies out there. Some are unbiased, some are not exactly single cell, some only can do a dozen markers. What about if we work as a community to start defining panels of genes in normal context as a core set that we could all agree upon per organ system as a core that would help with alignment, with context, and then every disease could tailor it. There's some thoughts here. There's opportunities for partnerships with industry in doing this that could help all of us align, get discounts. There's people in the room from industry, so sorry putting it out there. Some food for thoughts. That could be organ specific. Some cells are shared. So where my immune bionetwork kept for a minute. What about we try to define some panels that could help us study biology across tissues, right? This is an example of minimal viable product. Leverage our version 1 atlas. We're doing community annotation, trying to derive consensus annotation using some platforms like CAP. Can we leverage this to start thinking about what are representative panels that we can use on different technologies to do this at scale? And then you tailor it to your like specific needs. To me, that's like a practical thing we could do in the next 2-3 years that would help for so many reasons, like alignment, integration, making it more cost effective, which means more tissues, and so food for thoughts.
In here, hi. I was just wondering for the panels, I think some of the company like 10x have done already this job with Xenium.
Well, I'll push back for a second, so I'll interrupt you while keep the mic, which is they've actually engaged with just a few key opinion leaders or KOL. So when myself, I come in and, with my different hats, I look at these panels, it's missing all my favorite markers.
Of course.
So what about we use some of the atlas we're building, which is more thorough, which is not built out of just 2-3 data sets, but actually across several data sets, and use the community to kind of have something that's a bit more thorough.
But this is, I think, a basis we can take, a start with.
Yes.
Because this work has been done, and I think they have done it with already teams that were experts in every organs.
Are you from 10x?
No, I'm not from 10x.
Sorry, I'm just, because I'm going to push back. So I wanted to see if you're from 10x.
No.
I disagree. But this is me, Chloe, not me, moderator, talking for a second. They did it with some teams, but it was definitely not through a proper... so I commend their effort, of course, and you need to start somewhere, and we should not disregard what has been done, of course. But they've done it for some tissues, and so then there's a whole question of should we just use unbiased panels? There's 1K, 5K, regardless of the technology. Well, it's not always scalable. It's not. We talk about equity. It's not everybody that can systematically run 5K panels. Very expensive. It takes a lot of time to run. It's also noisy in some sense, and so there's not a one-size-fits-all solution. But how can we help empower a larger community where people will see the panel and say like, oh, I can use this as a core, let me modify it, and like you use that as a core, and then you can always add 50 or 100 genes to it.
Exactly. And I was just, sorry, if I can just add on that point, for the panels that we could define, we could use the single cell RNA-seq that we already have been generated and the complete version 1 or version 2 that DHA has already generated, and use any of the pipeline that help to locate, to use this as a reference as well, and panel for each cell type, organ specifically. Yeah, definitely.
Yeah, and that's exactly kind of the concept behind that. You have a minimal set, which everybody agrees to use. We are all scientists. We always will disagree, right? That's who we are. But there is a minimum we all agree to use so that we can map. And again, think of, how we can leverage outside of your narrow scope. Is there elements where we see the fibroblasts or T cells or fill in the blank of your underlying biology you are interested in, which also then allow you to walk across organs and walk into organs we even don't have on our panels yet. And that's a piece which allows you to reach out and to reach out particularly in under-resourced environments, because if we do that, then suddenly if you have a study of 15 patients in your disease of interest, you have 1,500 patients from 5 continents where your signal suddenly becomes very meaningful if it's similar or particularly if it's not. But that requires that we develop an approach how to really get representation and not by just talking to some of the key people.
So are you saying you kind of agree with what I was saying?
Absolutely.
Okay.
but just again, forcing the mind, think outside of your box if at all possible. That's what makes Human Cell Atlas so unique, and there are incredible opportunities ahead of us. Challenges with the spatial technologies, they are very, very heterogeneous. So if we don't do that, we will have a jungle out there, and nothing will map into anything.
I couldn't agree. And just before we move there, I'm going to add a comment, like we're talking in the context of a specific technology, but here what I'm talking about, and I think my thesis is alluding to, is leveraging the Atlas Assembly that has huge community involvement to define core sets of genes that could be applied to different technologies, which would also help alignment, whether you go with, I'm making it up, but Bruker or Merscope or 10x, and if we have some core sets of genes per organ system, I mean, already aligning across technology is going to be challenging. Aligning within technology is challenging. But agreeing on core sets of genes, well, could help move the needle.
And I think just to add that it's, I mean, in the group of Julio César Rodriguez, we have worked for years in trying to define functional signatures of pathways or transcription factors, right? And now moving to single cells, we realize that all of these are, well, we knew, right? But we can measure that these are context specific. So while defining panels is indeed a major endeavor and necessary to move to spatial, it's basically hard to judge, depending on the organ, which pathways do we measure, which genes, because KEGG focuses on members. Shall we look at downstream? We do not have also like lots of information about the effects. Like, just to give you an example, we are working with the cytokine dictionary and cytosig, and when you benchmark the activities of known cytokines, you observe that they are completely different depending on the cell line, depending on the concentration, depending on every context, right? So I agree, this is a major endeavor, and itself, I wouldn't say that it's as pragmatic, right? It's in itself an extremely hard task without the right data, which would be, is the HCA would, like, for example, get engaged in developing like large perturbational screens with known, like, I don't know, cytokines, right? Why don't we just generate a cytokine dictionary as an HCA?
Yeah, I think that can definitely be an example of an opportunity. And I think it's important also to think in terms of both, again, I'll bring us back to commonalities and versioning. And also when you talk about perturbation, we're back in the lens of disease, where you need a lot of information to develop these tailored panels. And to me, that's like a separate context where you need a lot of data. Here I'm talking about, well, even if you want to study disease, you still need to know at its core what are the cells you're supposed to have in a specific organ. At steady state in the heart, what is supposed to be there, what you would want to know if it changed. And whether you study, well, I'm not a heart expert, so my examples are going to be horrible. Viral myocarditis or congestive heart failure, I need help. At the end, there might be like a core set of markers of cells, you know, defining your cardiomyocytes, your macrophage, and it's some nerves and muscle. I'm going to stop there because I'm not a heart person. Like these you would want to measure regardless. And so I'm talking about like defining this core set. And then if you're going to study your favorite disease, then a specific group should think about these additional data sets that are needed to kind of tailor it.
And there will be a commitment, right? Because we cannot target the 150 cell states that we are aiming at every like atlas, right? We at some stage we would need to say like, okay, given the current technologies, we may just take 10.
Well, that's part of the conversation we should be having as we try to build a reference atlas of a healthy, which God knows how to define healthy, but normal tissue.
We can start just with the cells that we know they are in every tissue, the fibroblasts, the blood vessel, the nerve, the immune cells. So the cells that we know are everywhere. So if you start from there, then you can use it in every tissue, health and disease, and you get your starting core.
And I completely agree about perturbation and pathways and where do you end. And then this is where perhaps there's different combinations of technologies. One can do like specific panels, and some technologies are not destructive. And so given that funding exists, one can do some more unbiased analysis on it through like some sequencing approach. I feel like you want to.
Chloe, can I make one comment about panels and equity very quickly, I think, towards the end? We've had a number of collaborations within our projects, India and Brazil being a key one. And there's not always been the ability, especially as we start to focus on a technology of interest, to have that technology represented in the country. And so some of the things we're talking about here, if for example, the clinical unit does not want to share the FFPE block with a US or another investigator, there's going to be a need for the industry partners for them to place this technology in site so they can generate their own data and have that equity to own their own data and be able to share it on their own will. And for us, we've actually lost collaborations. Vietnam was one of them because there was not an ability for us to find a common ground in which to both either share or the technology didn't exist in the country. And so as we think about who we want to partner with, where that technology exists regionally, Africa, Asia, even down in some of the global south, is going to be very important for us to think about equity and engagement scalability the same.
All right, so I think going back to the panels, and then even within the panels, say 500 genes, being able to tier it, back to her point, like, you know, there might be some low-hanging fruit, and then you can tier the priorities, whether it's like a 5, 10, 20, 100, 500 gene panel. 3 comments, and then we're going to wrap up because we're at time.
Just really quickly, I wanted to talk about something that would take forever. But I was initially, Chloe, after hearing the technologies presentation this afternoon, I was just so blown away. And then Kevin, what you said about, like, as you change data modalities, that all of a sudden what we thought was our interpretation of this transcriptomic profile is completely contradicted by the proteomic profile. Down limiting to certain markers is just like not that appealing to me. On the other hand, having an infrastructure for us to have in a unified way, in a very contextualizable way, biomarkers for X, and then so that we have across the board, cross tissue infrastructure for collecting our like really important biomarkers. I've done a total PubMed data mining for biomarker and gene, and then with a phrase that's after that, and what you end up with is semi-valuable, but it's nowhere in context to where we're at in single cell omics world right now. It's not adequate for that task. That doing all of PubMed is not adequate for that task. So, but on the other hand, if we had a way to collect everybody's best thoughts about cell type specific function states and disease markers and common effector biological processes of disease, that then we could collect that in a unified way, that would be very useful. And that could be used for panels or used for changing technology mode, and then confirmed towards it.
This is actually very helpful. So in terms of next up, because we are at time and want to give a chance for the keynote speaker to set up, what we'll do is we'll summarize all of these key discussion points. I mean, we heard a lot of this recurring theme about measurements that are easy to do systematically, infrastructure, metadata, trying to kind of think about panels. Like, do we take a disease? Do we take all disease comers? Is it a mix? We'll summarize all of this, and we'll briefly present it. And then we'll follow up with a few outreach activities so we can continue this discussion. Our goal between now and December is to try to kind of define not only the big question, but one of the most burning priorities where we can immediately do something about it. So infrastructure is one of them. Metadata is one of them. Making recommendations one of them. These panels are definitely something we can work on and ask for more feedback. And then our goal is to try to kind of align funders and do fundraising to try to empower this. And this is about mapping the next decade. So we're not solving this now. And so we're going to continue this conversation. And we really want all of you to engage. You get a chance to help us shape it, right? It's not a few of us shaping it. It's all of us shaping it. Tackling disease is too unbounded. So we need to try to think about how we're approaching this. Thank you for our 2 speakers today for engagement. Grace, Kevin, any thoughts? No?